Hello, dear student. Welcome to the Sanjivani Mahavidyalaya Chapoli, affiliated to Swami Ramananda Tirth Marathwada University, Nanded. Here is Department of Botany, semester 4th and 6th, paper 9th and 14, plant metabolism, biochemistry and biotechnology for second year and third year student. In unit first, we discuss about photosynthesis. In previous lecture, we see the photosynthetic pigment, different types of photosynthetic pigments that are helpful in the photosynthesis process. Now you know, or it is clear that the photosynthetic pigments that are absorb light, and by using that light energy, the chemical energy is produced. In this lecture we discuss about the absorption spectrum. The photosynthetic pigments are capable for absorbing different wavelengths of light. When amount of light absorbed by a pigment is plotted as a function of wavelength, we obtain absorption spectrum. When the amount of light absorbed by a pigment is plotted as a function of wavelength, we obtain absorption spectrum. And that absorption spectrum varies from pigment to pigment by passing light of specific wavelength through a solution and measuring the fraction absorbed, we obtain the absorption spectrum of that medium or substance. Each type of molecule have a characteristic absorption spectrum, and measuring the absorption spectrum can be useful to identifying some unknown substances isolated from a plant or animal cell. What is absorption spectrum? The photosynthetic organisms contain one or more photosynthetic pigments that photosynthetic pigments are capable of absorbing different wavelengths of light. When amount of light absorbed by a pigment or a substance is plotted as a function of wavelength, you then find out the absorption spectrum or obtain the absorption spectrum. And that absorption spectrum varies from pigment to pigment. It means each pigment or every pigment have characteristic wavelength of light is absorbed. And that absorption spectrum is different for different pigments. Then we will discuss about the action spectrum. It represent action spectrum represent or extent of response. It represent extent of response to different wavelength of light in photosynthesis. It can also be defined as a measure of process of photosynthesis when a light of different wavelength is supplied, but the intensity is the same. For photochemical reactions involving single pigment, the action spectrum has same general shape as the absorption spectrum of that pigment, otherwise both are quite distinct. Quantum requirement and quantum yield. The solar light that comes to earth in the form of small packets of energy, that is known as photons, and energy associated with the photons is called quantum energy. This requirement of solar light by a plant is measured in terms of number of photons or quanta. The number of photons or quanta required by a plant or a leaf to release one molecule of oxygen during photosynthesis is called quantum requirement. How many photons is required for release of one molecule of oxygen is known as quantum requirement. It has been observed that in most of the cases the quantum requirement is 8. It means that 8 photons or quantums are required to release one molecule of oxygen. The number of oxygen molecules released per photon of light during photosynthesis is called quantum yield. The number of oxygen molecules released per photon is called quantum yield, and the quantum, if the quantum requirement is then 8, then the quantum yield will be 0.125, that is 1 by 8. Photosynthetic unit or quantazome. It is defined as the smallest group of collaborating pigment molecules necessary to affect the photochemical act. Quantazome or photosynthetic pigment. It is defined as the smallest group of collaborating pigment molecules necessary to affect the photochemical act, that is absorption and migration of light quantum to trapping center where it promotes the release of an electron. Emerson and Arnold in 1932, on the basis of certain experiments, assumed that about 250 chlorophyll molecules are required to fix one molecule of carbon dioxide in photosynthesis. 250 chlorophyll molecules are required to fix one molecule of carbon dioxide in photosynthesis. This number of chlorophyll molecules was called the chlorophyll unit. 250 molecules of chlorophyll is called as chlorophyll unit, but the name was subsequently changed to photosynthetic unit, and later it was designated as quantazome by Park and begins in 1964. The size of quantazome is about 18 into 16 into 10 nanometer and found in the member of thylakoids. Each quantazome consists 200 to 240 chlorophyll, that is 160 chlorophyll A and 70 to 80 chlorophyll B, 48 carotenoids, 46 quinone, 116 phospholipids, 144 digalactosyl diglyceride, 346 monogalactosyl diglyceride, 48 sulfolipids and some sterols and special chlorophyll molecules, that is P680 and P700. P is the pigment. 680 and 700 denotes the wavelength of this light molecule absorbed. So P680 and P700 constitute the reaction center or photocenter. Other accessory pigments and chlorophyll molecules are light gatherer or antenna molecules. It captures solar energy and transfers it to the reaction center by resonance transfer or inductive resonance. Photoluminescence. It is the phenomenon of re-radiation of absorbed energy. It is the phenomenon of re-radiation of absorbed energy. It is of two types, fluorescence and photorescence. The normal state of molecule is called as a ground state or subject state, a singlet state. When an electron of a molecule absorbs a quantum of light, it is raised to higher level of energy, a state of or excited second singlet state. From first singlet state, excited electron may return to the ground state either losing its extra energy in the form of heat or by losing energy in the form of radiant energy. The latter process is called fluorescence. When the electron molecule when an electron of a molecule absorbs quantum light, when electron absorb light, the electron in excited state, it is raised to into higher level, energy level, and that is called second singlet state. From first singlet state, excited electron may return to ground state either losing energy in the form of heat or losing energy in the form of radiant energy. That process is called fluorescence. The substance which can emit back the absorbed radiation is called fluorescent substance. All photosynthetic pigments have the property of fluorescence. The excited molecule also loses its electronic excitation energy by internal conversion and comes to another excited state is called triplet state. From this triplet state, excited molecule may return to ground state in three ways: by losing its extra energy in the form of heat, by losing extra energy in the form of radiant energy is called phosphorescence. The electron carrying extra energy may be expelled from the molecule and it consumed in some other chemical reaction, and a fresh normal electron returns to the molecule. This mechanism happens in chlorophyll a universal photosynthetic pigment. So, dear students, here we stop, and in next period or lecture we discuss about the immersion ray drop effect and enhancement effect. Thank you.